Hey dreamers, night owls, and tomorrow chasers. Welcome to Hafee Sleep. Ever find yourself scrolling before bed, wondering what the future holds? Maybe even how long that future could last? Well, tonight, we're diving into one of humanity's oldest dreams, and it's no longer sci-fi. Scientists are right now reversing aging in mice, growing human organs in labs, and rewriting what growing old even means. This isn't just possible, it's closer than you think. But before we unlock the secrets of longer, healthier lives, I need you with us. If you love peeking into the future, hit that subscribe button and smash like. It fuels our mission to explore what's next, together. Oh, and drop a comment below. Tell me, where in the world are you watching? Is it late night curiosity or early morning inspiration? Let's see how far our Hafee Sleep family stretches. Ready to reimagine the human lifespan? Let's begin. Imagine waking up not just feeling rested, but feeling young. Not just good for your age, but genuinely vibrant, energetic, like the best version of yourself in your prime. Now imagine that feeling stretching out, not just for decades, but perhaps for centuries. Sounds like science fiction, right? Something out of a dusty old book about mythical fountains or alchemists' potions. Well, buckle up, because what was once pure fantasy is now knocking firmly on the laboratory door. Scientists are telling us, with growing confidence, that within our lifetimes, perhaps by the middle of this very century, some form of significant life extension could become widely available. Think about that. For the first time in human history, we might genuinely have a shot at turning the tide on aging itself. It all starts, strangely enough, with mice, little creatures scurrying in cages under the watchful eyes of researchers. And in these labs, something truly astonishing is happening. Scientists aren't just slowing aging down in these mice, they're actually reversing it. Wrinkles smooth out, fur regains its youthful sheen, energy levels surge, and damaged organs seem to repair themselves. How? It's not magic. It's about giving their DNA repair crews a serious upgrade. You see, inside every single one of your trillions of cells is your DNA, the intricate instruction manual for building and maintaining you. But just like any manual that gets used constantly, pages tear, ink fades, errors creep in. Over time, these errors accumulate. Our cells have built-in repair teams, molecular mechanics constantly fixing typos and patching up breaks. But as we age, these repair crews get sloppy, overwhelmed, or just plain tired. They miss things. And those missed repairs? That's the fundamental damage that is aging. In one of our earlier science stories about anti-aging, I have explained all these things in details. What these scientists did was find ways to rejuvenate those repair crews in the mice. They used clever techniques like tweaking specific genes or introducing special molecules to essentially give the repair mechanisms a potent shot of espresso. Suddenly, the crews were working like they did in the mouse's youth, fixing DNA damage more efficiently. The result? The biological clock wasn't just slowed, it was wound backwards. Aged mice became biologically younger. Now, translating that precisely to humans is a massive leap. We're infinitely more complex, but the principle is revolutionary. It proves that aging isn't some immutable one-way street. It's a biological process, and potentially a treatable one. But life extension not just about fixing what's inside. Let's be honest, even the best-tuned engine, running perfectly for thousands upon thousands of hours, eventually sees parts just wear out. Think about your faithful old car. You change the oil religiously, keep the spark plugs clean, maybe even upgrade the fuel injector. The engine purrs like a kitten. But what about the transmission? After shifting gears countless times, those gears grind down, the fluid gets tired, the synchros start to complain, or the suspension. Years of bumps and potholes take their toll. You can't tune up everything forever. Eventually, you need a replacement part. Our bodies, same deal, just infinitely more complex and precious. Even if we manage to keep the cells themselves youthful and buzzing with energy, the grand structures they build, our organs, face monumental physical challenges over decades and hopefully soon centuries. Imagine your kidneys. These incredible biological filters work 24/7, sifting through your entire blood supply multiple times a day, meticulously pulling out toxins and waste products, balancing fluids and salts. Picture them as two dedicated factory workers processing a never-ending river of liquid. After, say, 120 years of continuous, non-stop filtering, wouldn't you expect some of the filtering mesh to get a bit clogged? Some of the tiny tubes to get a bit scuffed? The sheer volume of material they've handled is staggering. Millions of gallons, trillions of toxins processed. It's not that the cells inside the kidney are necessarily senile zombies. It's that the entire organ, the intricate plumbing system, the delicate filtration membranes, have endured incredible physical stress. Like a beloved, well-used teapot, the material itself just gets thin in places. Or consider your heart. That tireless muscle beats and beats and beats, roughly 100,000 times every single day. Over 80 years, that's nearly 3 billion beats. Imagine flexing any other muscle in your body 3 billion times without a break. The valves that keep the blood flowing in the right direction, they're like tiny, perfect doors slamming shut with every beat, billions of times. Even the most resilient biological materials can develop tiny tears, calcifications, or just lose a bit of their springiness with that kind of relentless use. The joints in your knees and hips, they're bearing your weight, absorbing impact, twisting and turning millions of times. The cartilage, that smooth, slippery cushion, inevitably wears thin. It's not necessarily faulty cells. It's physics and time taking their toll on the macroscopic structure. So cellular rejuvenation is vital, the ultimate foundation. It's like having a factory full of young, energetic, perfectly trained workers. But if the factory building itself is crumbling, the machinery is worn out, and the conveyor belts are fraying, even the best workers can only do so much. This, my friends, is where the story takes a truly breathtaking turn, into a field that sounds like pure science fiction, but is happening right now in laboratories around the globe. Regenerative medicine. Specifically, the audacious, magnificent goal of growing entirely new custom-made organs in the lab. Yes, you heard that right. We're not just talking about patching things up or stitching bits together. We're talking about fabricating a living, functioning kidney, or a liver, or a lung, or that tired old heart, from scratch. Well, almost from scratch. The magic ingredient, the biological spark that makes this possible, is something you might have heard of. Stem cells. Think of stem cells as the ultimate raw material, the master builders, the blank slates of the biological world. They're like the unformed clay before the sculptor begins. Most cells in your body are specialists. A heart muscle cell is brilliant at contracting, but it has no clue how to be a brain cell. A skin cell is a fantastic barrier, but it can't filter blood like a kidney cell. They're locked into their specific jobs. Stem cells, however, are different. They are the originals, the pluripotent powerhouses. Pluripotent just means they have the potential to become almost any type of cell in the body, given the right instructions. They hold within them the blueprint for building a whole new you, or at least a new part of you. Where do we get these wonder cells? There are a few ways, each with its own fascinating story. One source is embryonic stem cells. These come from very, very early stage embryos, usually donated from IVF clinics with consent, specifically from embryos that wouldn't be used otherwise. These cells are incredibly powerful, truly blank slates with maximum potential. But their use has ethical considerations. And also, if you grew an organ from someone else's embryonic stem cells and put it into you, your immune system would likely recognize it as foreign and attack it. Rejection City. This is where the real revolution for personalized organs came in. Induced pluripotent stem cells, or iPSCs. This discovery, which earned Shinya Yamanaka a Nobel Prize, is nothing short of genius. Imagine taking a tiny piece of your skin, maybe from your arm, just a few cells, nothing dramatic. Scientists then take these ordinary, everyday skin cells, which are specialists, remember, and perform a kind of biological alchemy. They introduce a specific cocktail of genes, often using harmless viruses as delivery trucks, or newer, safer methods that essentially rewind the skin cells back in time. Zap. Those skin cells forget they were skin cells. They revert back to a state almost identical to those powerful embryonic stem cells. They become your pluripotent stem cells, carrying your unique genetic blueprint. It's like taking a brick from your house and magically turning it back into clay that can be molded into any other part of a house, a window pane, a roof tile, a piece of plumbing. And because they're made from your cells, any organ grown from them would be genetically yours. Your immune system would see it and say, "Ah, friendly, welcome back. No rejection drugs needed." That's the dream. So we have the magical clay, the iPSCs, made from your skin. Now, how do we shape that clay into a complex, three-dimensional, working organ? This isn't like molding a simple bowl. A kidney has millions of precisely arranged cells of different types. Filtering cells, tube cells, structural cells, intricate blood vessel networks, nerves, and it all has to fit together perfectly to function. It's mind-bogglingly complex. Here's where the lab becomes a biological workshop, employing some truly ingenious strategies. The most common approach involves something called a scaffold. Imagine you're trying to build a magnificent, elaborate sandcastle. You don't just dump sand and hope for the best. You might use a bucket to form the main tower, carefully carve out arches with a small tool. That bucket, that tool, they're your scaffold. They provide the shape, the structure, the boundaries within which the sand, your cells, can be organized. In the lab, scientists create artificial scaffolds designed to mimic the exact shape and microscopic architecture of the organ they want to grow. How? Sometimes they use incredibly detailed 3D scans of a real, healthy organ, maybe from a donor, or even a scan of your organ if it's not too damaged. Using this digital blueprint, they can employ advanced 3D printing techniques, but instead of printing with plastic or metal, they print with special biomaterials. These are carefully engineered materials designed to be biocompatible, meaning your body won't freak out and reject them, and often biodegradable. Think of materials like collagen, a major component of your natural tissues, or specially formulated gels that feel like a soft, supportive jelly. The printer lays down this material, layer by minuscule layer, building up a perfect, intricate replica of the organ shape, complete with all the tiny channels and cavities where blood vessels or urine-collecting tubes need to go. It might look like a ghostly, translucent version of a kidney or a heart, made of this spongy, gel-like material. This scaffold provides the crucial wear for the cells. Now comes the seeding. Scientists take that precious suspension of your iPSCs, millions of them, swimming in a nutrient-rich broth, and carefully introduce them onto this scaffold. It's like gently sprinkling seeds onto perfectly prepared soil. But we don't just want stem cells. We need them to turn into the right kinds of cells in the right places. This is where the real artistry of developmental biology comes in. Remember, stem cells become specialists based on the signals they receive. It's like training. The scaffold is placed inside a sophisticated bioreactor. Think of this not as a reactor for nuclear fusion, but as a high-tech womb. It's a sterile chamber that provides the perfect, nurturing environment. It controls the temperature precisely, body temperature, of course. It pumps in a constant flow of that nutrient-rich broth, mimicking blood flow, bringing food and oxygen to the cells and washing away waste. And crucially, scientists introduce very specific signaling molecules into this broth. These are like biological instructions, whispers telling the stem cells what to become and where to go. How does this work? It's based on how nature builds organs in the first place, during embryonic development. Scientists have painstakingly decoded the chemical conversations that happen when cells are deciding their fate. Need cells in one area to become the delicate filtering units of a kidney, nephrons. They might add a specific growth factor, like BMP7 or FGF2, at just the right concentration and time. Need the cells lining the scaffold's channels to become smooth muscle cells to help the ureter contract. They might add TGF-beta. Need to encourage blood vessel formation? They introduce VEGF, vascular endothelial growth factor. It's an incredibly complex, timed symphony of chemical signals, carefully orchestrated by the scientists. Over days, weeks, and sometimes months, something remarkable happens within that bioreactor. The stem cells, bathed in this precisely tuned cocktail of signals, start to change. They differentiate. Some, receiving specific instructions, transform into kidney tubule cells. Others, in a different location receiving different signals, become the cells that form the intricate glomerular filters. Others still become the cells that build the connective tissue framework. It's not random. Guided by the scaffold's shape and the chemical gradients, they start to organize themselves. They multiply. They migrate to their designated spots. They begin to connect to each other, forming the complex networks essential for function. It's like watching a bustling city being built from the ground up by millions of microscopic workers who know exactly where to put the roads, the power lines, the water pipes, and the buildings. They're literally self-assembling, guided by the blueprint provided by the scaffold and the instructions from the chemical signals. But wait, there's a massive challenge here, especially for thick, complex organs like the heart, liver, or kidney: vascularization. An organ isn't just a lump of cells. It needs a dense, branching network of blood vessels, capillaries, arterioles, venules, to deliver oxygen and nutrients deep into every nook and cranny and carry away waste. Without this, cells deeper than a fraction of a millimeter would suffocate and die. Growing this intricate, life-sustaining highway system inside the developing organ is one of the biggest hurdles in regenerative medicine. Scientists are tackling this in several clever ways. Designing smarter scaffolds. They're creating scaffolds with built-in, incredibly fine channels using advanced 3D bioprinting techniques. These channels are designed to precisely mimic the branching patterns of natural blood vessels. The hope is that when they seed endothelial cells, the cells that line blood vessels, into these channels, they'll naturally form tubes along the predefined paths. It's like laying down empty pipes and hoping the plumber cells will come and seal them perfectly. Co-seeding cells. Instead of just seeding stem cells, scientists might also introduce endothelial cells and their support cells, parasites, right alongside the organ-specific cells right from the start. The idea is that as the organ tissue forms, these vascular cells will simultaneously start weaving their own network within it, encouraged by growth factors like VEGF. It's like having the road crew and the building crew working side by side from day one, using nature's blueprint: decellularization, recellularization. This is a fascinating and slightly eerie approach. Scientists take a real organ, perhaps from a pig, which are surprisingly similar to human organs in size and structure, or even a human donor organ that wasn't suitable for transplant. Then, using special detergents and washes, they gently strip away all the original cells. What's left is the organ's natural scaffold, the intricate extracellular matrix, a complex web of collagen, proteins, and sugars that held the cells in place, complete with the perfect natural architecture of blood vessels, airways, or ducts. It's like a ghost organ, the perfect detailed shape, but empty. Then, they take your iPSCs, differentiate them into the right cell types, and seed them back onto this perfect natural scaffold. The cells crawl into their natural niches, guided by the biological signals still present in the matrix, and ideally repopulate the entire structure, including the blood vessel linings. It's like moving into a perfectly preserved, biologically compatible house, where all the plumbing and wiring are already in place. So, let's see this in action with something scientists have already achieved: lab-grown windpipes, tracheas. The trachea is a tube, relatively simple in structure compared to a kidney or heart, but vital for breathing. Patients with severe damage from cancer or injury faced grim prospects. Here's how regenerative medicine offered hope. 1. The scaffold. A donor trachea, human or sometimes animal, was decellularized, stripped of its cells, leaving the hollow tubular scaffold of connective tissue. 2. The cells. Stem cells, often mesenchymal stem cells, which can become bone, cartilage, and fat cells, harvested from the patient's own bone marrow, were taken. 3. Seeding and maturation. These stem cells were painted onto the inside and outside of the scaffold and then placed in a bioreactor. Inside the bioreactor, bathed in nutrients and specific growth factors, the stem cells differentiated. Some became the cartilage rings that give the trachea its structure and prevent collapse. Others became the ciliated epithelial cells that line the inside. These are crucial because they have tiny hairs that beat rhythmically to sweep mucus and debris out of the airways. They learned their jobs on the scaffold. 4. Transplant. Once matured, this new trachea, built on a donor scaffold but covered in the patient's own cells, was transplanted. Because the outer cellular layer was the patient's own, the risk of rejection was dramatically lower. Patients received a functional airway. While there have been challenges and complexities, these pioneering procedures proved the concept. We can build complex biological structures. Bladders have also been successfully engineered and implanted in patients, particularly children with spina bifida who had poorly functioning bladders. In this case, 1. Biopsy. A small piece of the patient's own bladder tissue was taken. 2. Cell culture. Muscle cells and urothelial cells, the lining cells, were separated and grown separately in the lab, multiplying millions of times. 3. Scaffold. A biodegradable bladder-shaped scaffold, often made of collagen or a synthetic polymer, was used. 4. Seating. The muscle cells were seated onto the outside of the scaffold and the urothelial cells onto the inside. 5. Maturation. The seated scaffold was incubated, allowing the cells to form layers. 6. Implantation. The new bladder patch or replacement was surgically attached to the patient's existing bladder structures. Over time, the scaffold dissolved, leaving a functioning bladder made primarily of the patient's own cells. Long-term studies show these engineered bladders can work well for years. Now, kidneys are a much tougher nut to crack. They're incredibly dense, with over 1 million tiny filtering units, nephrons, per kidney, each a miniature chemical processing plant with multiple components. Growing a whole, fully functional, transplantable human kidney in a lab is still on the horizon, but progress is breathtaking. Kidney organoids. Scientists are masterful at growing miniature kidney organoids from stem cells. These aren't full-sized kidneys, but tiny, simplified versions, a few millimeters across, that contain many of the key cell types and structures arranged correctly: glomeruli, tubules, even early blood vessel cells. They can even filter blood-like fluids in lab tests. These organoids are invaluable research tools. Scientists use them to study kidney development, test drugs for toxicity, and understand kidney diseases in ways impossible with flat cell cultures. They're the stepping stones. Building blocks. Researchers are focusing on growing larger, functional pieces. Imagine successfully bioengineering a perfect, dense patch of nephrons, or a perfectly formed ureter, the tube draining urine, or mastering the vascular network. The goal is to eventually assemble these perfected modules into a whole organ. Think advanced Lego bricks for life. Decellularized kidneys. Progress is being made in repopulating decellularized pig or human kidney scaffolds with human stem cells. Getting cells to populate the entire dense structure, especially the delicate filters, and connect properly is hugely complex. But each experiment teaches us more. Seeing human cells begin to function within this natural architecture is incredibly promising. And let's not forget the heart. Engineering heart muscle patches is a major focus. Imagine someone suffers a heart attack. A chunk of their heart muscle dies, replaced by scar tissue that doesn't beat. Scientists are growing small, beating patches of heart muscle, derived from iPSCs, in the lab. The goal is to surgically stitch these living, pulsating patches directly onto the damaged area of the patient's heart. These patches wouldn't just sit there. They'd integrate with the existing heart tissue, beat in sync, and actually contribute to pumping blood. Early animal trials show this can improve heart function. While a full bioengineered heart transplant is far more complex, it needs valves, intricate electrical conduction systems, perfect strength and durability. These patches are a revolutionary step. They demonstrate we can create functional beating heart tissue that integrates. The technology advancing all this? 3D bioprinting is rapidly evolving beyond just making scaffolds. Imagine a printer, but instead of ink, its cartridges hold bioinks. These aren't just gels. They can contain living cells, growth factors, and supportive materials. The printer head moves with incredible precision, depositing tiny droplets or fine strands of these bioinks layer by layer, following a digital 3D model. The result? It's directly printing complex living structures. Early successes include printing skin grafts for burn victims, layering different skin cell types, relatively simple tissues like cartilage for nose or ear reconstruction, already used experimentally, and even intricate structures like liver lobules or sections of heart tissue with built-in microchannels. The dream is to one day bioprint entire organs directly, cell by cell, structure by structure, with the blood vessels pre-integrated. We're not there yet for complex organs, but the trajectory is clear. So, let's bring this back to our original vision, that future we're sprinting towards. Imagine it's the year, let's say 2150. You've benefited from a lifetime of cellular tune-ups, maybe regular senolytic treatments clearing out your zombie cells, epigenetic resets, keeping your gene expression youthful, NAD+ boosters energizing your mitochondria. You feel fantastic, mentally sharp, physically strong. But let's face it, you've been around for 150 years. That original heart of yours, despite all the internal cellular care, has simply performed its heroic duty of billions upon billions of beats. The valves are a bit less supple, the muscle fibers a tad less springy. It's not failing catastrophically, but it's tired. Like a well-loved vintage watch that keeps good time but needs a significant overhaul. No problem. Decades ago, when you turned 100, a small skin biopsy was taken. Just a pinprick. Your cells were used to create your personal bank of iPSCs. Now, your medical team calls up your digital file, using incredibly detailed scans of your current heart to get the perfect size and shape, maybe even accounting for any unique quirks. They design the blueprint. A sophisticated bioprinter, or perhaps a team of bioengineers using a hybrid scaffold approach, begins the meticulous process. Your iPSCs are awakened and directed, step by step, to become cardiomyocytes, heart muscle cells, valve cells, endothelial cells for the blood vessels, pacemaker cells, the whole orchestra. They're nurtured in a bioreactor that mimics the pressures and flows of the real circulatory system, strengthening the new tissue. Signals coax them to form the dense, aligned muscle fibers needed for powerful contractions and the intricate electrical pathways that keep the beat perfectly synchronized. After several weeks, your new heart is ready. It's not cold metal and plastic. It's a warm, living, beating organ, pulsing gently in its sterile chamber. Genetically, it's you. The surgery is still major, of course, but incredibly precise, guided by real-time imaging. The old, faithful heart is respectfully retired. The new one is connected. Your body recognizes it instantly as self. No rejection drugs needed for life. As it integrates, powered by your robust, rejuvenated cellular machinery elsewhere, it takes over its vital rhythm. The clock on that particular organ, reset, 0 miles, brand new. This isn't just about hearts, that kidney struggling after a century and a half of filtration duty. Swapped for a new one, grown to order. Hips worn thin from centuries of walking. Replaced with lab-grown joints using your own cartilage cells. Maybe even sections of liver, pancreas, or lung can be replaced as needed. The deteriorating effects of aging transform from an inevitable, terrifying decline into a series of manageable maintenance issues, like taking your classic car into the shop for a transmission rebuild or a new suspension. Complex, yes, but routine. The difference? The parts aren't manufactured in a factory. They're grown in a lab, custom-made from your own biological essence. This future, powered by the twin engines of cellular rejuvenation and organ regeneration, isn't about chasing immortality in a fragile, decaying shell. It's about achieving longevity escape velocity. In the truest sense, not just adding years to life, but adding vibrant, healthy, functional life to years. It's about compressing the period of frailty, pain, and dependency into a vanishingly small sliver at the very end, or perhaps eliminating it altogether. It's about maintaining the physical vessel so the mind and spirit within can continue to explore, learn, love, and create for durations we can barely imagine today. The worn-out transmission is replaced. The tired suspension is renewed. The engine, meticulously maintained, keeps on purring. The journey continues, long and full of life. That's the breathtaking promise growing, quite literally, in labs right now. So, why now? Why, after centuries of dreaming about fountains of youth and philosophers' stones, are serious scientists in crisp white lab coats standing up and saying, with straight faces, significant life extension? Yeah, that could be on the menu within a few decades. It's a fantastic question. It feels sudden, doesn't it? Like we went from hoping aspirin might help us feel a bit better to talking about resetting our biological clocks. But this surge of optimism, this feeling that we're genuinely on the cusp of something monumental, isn't magic. It's the beautiful, messy collision of two powerful forces: relentless human desire, meeting unprecedented scientific capability. Let me unpack that. First, the desire. Oh, that's as old as humanity itself. It's etched into our oldest stories. Gilgamesh trekking to the bottom of the sea for the plant of immortality. Chinese emperors sending fleets to find Penglai, the mythical island of the immortals. Spanish explorers hacking through jungles searching for El Dorado and its rumored rejuvenating waters. Alchemists in smoky labs across medieval Europe, desperately trying to brew the elixir of life from mercury and ground-up gems. Yikes. Even Ponce de Leon traipsing around Florida looking for that legendary fountain of youth. It's everywhere. In our religions promising eternal life, in our fairy tales with witches granting long life, often with a nasty catch, in the sheer gut-wrenching fear of losing loved ones or facing our own decline. It's fundamental. Who wouldn't want more time? More sunrises? More laughter with family? More chances to learn that language, climb that mountain, write that book, hold a great-great-grandchild? It's the deepest, most human yearning, the desire for more life, lived fully and vibrantly. For millennia, this was pure, agonizing, wishful thinking, powerful, driving, but ultimately frustrating. We could dream it, sing about it, write epic poems about it, but we couldn't do anything about it. Biology felt like an immutable sentence. But then, science started catching up, not with a single eureka moment, but with 1,000 tiny breakthroughs, slowly, steadily building over the last century, accelerating like a rocket in the last 20 years. It's like we finally gathered all the right tools, the right blueprints, and the right team, all at the same time. This isn't one field having a good day. It's a grand convergence. Imagine 5 mighty rivers, each carving its own path for years, suddenly surging together into one unstoppable torrent. That's what's happening right now in labs across the globe. Let's wade into those rivers. The genomic revolution, cracking life's operating system. Think of your DNA as the most complex, intricate instruction manual ever written. It's not just a blueprint. It's the master plan for building and running you, down to the tiniest molecular widget. For most of history, this manual was locked in a vault, written in a code we couldn't decipher. Then came the Human Genome Project. It was like a global scientific moonshot, Starting in 1990, thousands of scientists from around the world worked together on an audacious goal: sequence the entire human genetic code, all 3 billion letters of it. It took over a decade and billions of dollars, but in 2003, they did it. They published the first complete draft of the human genome. This was huge. Suddenly, the vault was open. We had the master plan. But it wasn't just a one-time read. The technology to sequence DNA, to read those billions of letters, started improving at a mind-blowing pace. Moore's Law? Forget it. DNA sequencing costs plummeted. Way faster than computer chips got cheaper. What cost billions and took years in 2003 can now be done for less than $1,000 in a single day. It's become almost routine. This means scientists can now read the fine print, compare the genomes of thousands of people who live exceptionally long, healthy lives, centenarians and supercentenarians, to those who don't. What tiny spelling differences, single letter changes called SNPs, SNPs, pop up more often in the long livers? These are clues pointing to genes that protect against aging or age-related diseases. Spot the typos. See exactly where damage accumulates in DNA as we age, the breaks, the mutations, the places where the repair crews missed a spot. We can pinpoint these hot spots of genomic instability. Find the repair crews. Identify the specific genes that code for all those molecular mechanics, the proteins like PARP1, DNA-PK, BRCA1-2, responsible for fixing DNA damage. We know their names, their addresses in the genome, and increasingly, how they work or fail over time. Edit the code, CRISPR. This is the game changer. CRISPR-Cas9 isn't just reading, it's editing. Think of it as molecular scissors and paste, guided by a GPS system. Scientists design a tiny piece of guide RNA that acts like a homing beacon, leading the CRISPR scissors, the Cas9 protein, to an exact spot in the genome. SNP. They can cut out a faulty gene, like one linked to a rapid aging disease. Or they can paste in a corrected version. Or even tweak the volume control promoters on a gene to make it produce more of a helpful repair protein. It's precise genetic surgery. While using CRISPR directly inside an adult human for anti-aging is still complex and risky, off-target cuts are a concern. It's revolutionary for research. Scientists can use CRISPR in cells or lab animals to rapidly test what happens if we boost this DNA repair gene or silence that one linked to senescence. It's accelerating discovery at an incredible rate. The blueprint isn't just readable, it's becoming editable. Stem cell mastery, unlocking the body's inner repair kit. Remember those stem cells we talked about for growing organs? Their story is central to why now. For decades, stem cells were biological ghosts, theorized, elusive, frustratingly hard to work with. Embryonic stem cells, ESCs, discovered in mice in the 1980s and humans in 1998, were powerful but ethically tricky and hard to control. Getting them to reliably become a specific cell type was like herding cats. Then came Shinya Yamanaka's Nobel-winning bombshell in 2006, induced pluripotent stem cells, iPSCs. This was pure biological alchemy. Yamanaka figured out that by adding just 4 specific genes, now often called the Yamanaka factors, OCT4, SOX2, KLF4, c-MYC, to an ordinary adult skin cell, he could reprogram it. That humble skin cell would forget it was skin. It would revert back to an embryonic-like state, a pluripotent stem cell. It was like turning a brick from your house back into universal clay. Suddenly, we had a source of powerful stem cells that were your own, made from your skin or blood, So no immune rejection issues for therapies. Avoided ethical issues. No embryos needed. Were plentiful. A tiny biopsy could yield billions of your own personalized stem cells, but the revolution didn't stop there. The past 15 years have seen an explosion in directing stem cells. Scientists have become master puppeteers, learning the intricate chemical and physical language that tells a stem cell what to become. Need heart muscle cells? Bathe them in activin A and BMP4 at precise times. Need dopamine-producing neurons for Parkinson's? Use a cocktail involving SHH and FGF8. Need kidney cells? Sequential doses of CHIR 99021 and FGF9 might do the trick. We're building vast libraries of these differentiation protocols. We've also gotten incredibly good at growing them at scale. Bioreactors that gently stir and nourish billions of stem cells, keeping them happy and multiplying. Purifying them. Separating the desired heart cells from the stray liver cells, for example. Building complexity. Moving from flat layers of cells in a dish to complex 3D structures, organoids and tissues that mimic real organs much better. Mini brains, mini guts, mini kidneys grown in labs are invaluable for testing drugs and understanding development and disease. This mastery isn't just for organ replacement. It's crucial for rejuvenation therapies too. Imagine harvesting your old tired cells, turning them back into iPSCs, fixing any accumulated DNA damage or epigenetic glitches while they're in their flexible state, then turning them back into youthful, healthy versions of the original cells and putting them back where they belong. It's like giving your body's workforce a factory reset and a tune-up. Advanced biotechnology, super senses and super tweezers. Science doesn't just need ideas, it needs tools. And the toolbox available to biologists today would make researchers from the 1980s weep with envy. It's like they were trying to fix a watch with a hammer and chisel, and now we have atomic force microscopes and laser-guided microscalpels. Let's geek out on a few. Seeing the invisible, cryo-EM super-resolution microscopy. How do you fix something if you can't see it? Cryo-electron microscopy, cryo-EM, flash freezes molecules in mid-action and blasts them with electrons, creating incredibly detailed 3D images of proteins, DNA complexes, even viruses, at near-atomic resolution. Super-resolution microscopy breaks the diffraction limit of light, letting scientists watch individual molecules move and interact inside living cells in real time. It's like having X-ray vision combined with super slow-mo for the nanoworld. We can see DNA repair happening, watch telomeres shorten, observe senescent cells spew their toxic brew. Molecular fishing manipulation. Tools like CRISPR, already mentioned, are just the tip. We have ways to grab onto specific proteins or RNA molecules and pull them out of cells to study them. Immunoprecipitation. We can use light, optogenetics, or chemicals to switch specific genes or pathways on and off with incredible precision inside living organisms, like flicking a light switch to see what happens. Imagine testing a potential anti-aging drug pathway by switching it on in just the liver of a mouse and seeing the effect. Rapid-fire DNA, RNA reading, next-gen sequencing. We touched on sequencing costs dropping, but the speed and throughput are equally revolutionary. We can sequence not just the genome, the static code, but the transcriptome, all the RNA messages being actively read from the genome, the epigenome, all those chemical tags controlling which genes are read, even the proteins being made, proteomics, for thousands of cells individually. This lets us see the dynamic state of cells and tissues in incredible detail as they age. Bioengineering marvels. Creating those intricate organ scaffolds, that's advanced biomaterials science and 3D printing. Designing bioreactors that perfectly mimic the pulsing flow of blood through a developing heart tissue, that's sophisticated fluid dynamics and engineering. Building tiny microfluidic labs on a chip to test drugs on miniature human tissue models, more cutting-edge engineering. The physical tools to build and manipulate biological systems are light years ahead. Computational power AI, taming the complexity beast. Here's the dirty little secret of biology. It's mind-bogglingly complex. Think about it. You have trillions of cells. Each cell has thousands of different proteins interacting in intricate networks. Genes talk to genes, pathways talk to pathways, organs talk to organs. Aging isn't one broken part. It's the gradual fraying of a million interconnected threads. Trying to understand this with pencil and paper, or even basic computers, is like trying to count every grain of sand on a beach using an abacus. Impossible. Enter supercomputers and artificial intelligence, AI. This is the indispensable brainpower behind the revolution, storing the tsunami of data. All that sequencing, all those microscope images, all the results from millions of lab experiments, we're generating zettabytes of biological data. That's a 1 followed by 21 zeros. Supercomputers provide the massive storage and processing muscle just to handle it. Finding patterns in the noise. AI, particularly machine learning, is brilliant at this. Feed an AI algorithm data from thousands of people, their genomes, their health records, their lifestyle factors, their biological age scores, and it can start finding incredibly subtle patterns that humans would miss. Maybe it spots that a combination of 3 specific genetic variants plus a certain gut microbiome signature plus elevated levels of a weird protein predicts rapid liver aging. Boom, a new target. Mapping the networks. AI helps build incredibly complex models of how all the aging pathways interact. How does tweaking nutrient sensing, like with rapamycin, affect senescent cell clearance? How does boosting NAD+ influence mitochondrial function and DNA repair? AI simulations can predict these interactions, suggesting the most promising combinations for therapies. Designing drugs at warp speed. Finding a drug that hits a specific aging target used to involve screening millions of chemicals in the lab, one by one, slow, expensive, like finding a needle in a haystack blindfolded. Now, AI can virtually screen-side billions of molecules in silico on the computer in days. It analyzes the 3D shape of the target protein thanks to tools like AlphaFold, another AI marvel that predicts protein structures and hunts for molecules that will fit into it perfectly, like a key in a lock, to activate or block it. It can even design completely new molecules predicted to work. This accelerates drug discovery for senolytics, autophagy boosters, epigenetic modulators, you name it, by orders of magnitude. Predicting outcomes. How will a specific patient respond to a potential anti-aging therapy? AI, trained on vast data sets, is getting better at making personalized predictions based on an individual's unique biology. Big data biomarkers, measuring the unseeable. You can't manage what you can't measure. For decades, measuring aging meant looking in the mirror, feeling creaky, or noting your chronological age. Hopelessly crude. The revolution here is twofold: gathering massive amounts of health data over long periods and developing precise molecular yardsticks for biological age. The mega-studies, biobanks. Imagine tracking 500,000 people for 20 years, recording everything: their DNA, their diet, exercise, sleep, environment, illnesses, medications, even their social connections. Then regularly taking blood, urine, maybe even images. That's the UK Biobank. Similar huge efforts exist worldwide, All of Us in the US, China Kadoorie Biobank, etc. These are treasure troves. Scientists can mine this data to find out what habits truly correlate with longevity, what early molecular signs predict heart disease or dementia decades later, what genetic or environmental factors accelerate aging. It's real-world evidence on a colossal scale. The molecular clocks, biomarkers of aging. This is where it gets really exciting. Scientists have discovered that our cells have molecular clocks that tick along somewhat independently of our birthday candles. The most famous are epigenetic clocks. Remember those chemical tags, methyl groups, stuck onto DNA that control gene activity? The pattern of these tags changes predictably as we age. By analyzing hundreds of thousands of these methylation sites in your blood or saliva, sophisticated algorithms, often AI-powered, can calculate your biological age. A much better indicator of your true cellular health and remaining lifespan than your chronological age. You might be 50 years old chronologically, but your biological age could be 45. Great. Or 60. Time for action. Other biomarkers include telomere length. Measured in white blood cells, shorter telomeres generally indicate older biological age. Senescent cell burden. Tests are emerging to estimate how many zombie cells are lurking in your tissues via specific proteins they secrete. Inflammation levels. Chronic low-grade inflammation, inflammaging, is a key driver. Blood tests for proteins like IL-6 or CRP are common indicators. Metabolite profiles. The levels of thousands of small molecules in your blood shift as you age, reflecting changes in metabolism. Mass spectrometry machines can read this complex signature. Proteomics, glycomics. Patterns of proteins and sugar molecules attached to proteins also change with age and disease. Why are these so crucial? They give us targets and feedback. Targets. Instead of vague fight aging, we can say, let's reduce this person's epigenetic age by 5 years, or clear 30% of their senescent cells, or lower their inflammatory marker X. Feedback. When you try an intervention, a new drug, a diet, an exercise regime, you can measure objectively and relatively quickly, months, not decades, if it's actually reversing biological age or improving key biomarkers. Is your epigenetic clock slowing down? Are your telomeres lengthening? Are inflammation markers dropping? This accelerates clinical trials massively. We don't have to wait 30 years to see if people live longer. We can see if the underlying biology is improving much sooner. It turns aging into a measurable, potentially treatable condition. This convergence didn't happen overnight. The idea that aging might not be set in stone has been simmering for a while. Let's take a quick jog through history. Ancient dreams. Early stabs. Alchemists, around 500–1500 AD, sought immortality through chemistry, mostly involving toxic metals. Not recommended. Explorers chased myths, 15th–18th centuries, not super scientific. The germ of an idea, 19th, early 20th century. Scientists like Elie Metchnikoff in early 1900s proposed that aging might stem from intoxication by gut bacteria, and that yogurt, full of good bacteria, could promote longevity. While overly simplistic, it was one of the first scientific theories linking cellular processes, gut microbes, to lifespan. He championed cellular health. the foundation stones around mid-20th century. This era laid the essential groundwork. DNA structure, 1953. Watson, Crick, Franklin, and Wilkins unravel the double helix. Suddenly, we understand the physical form of the genetic code. Revolution number 1. The Hayflick limit, 1961. Leonard Hayflick discovers that normal human cells in a lab dish don't divide forever. They stop after about 50 divisions. This introduced the concept of cellular senescence, cells hitting a hard limit. It suggested aging might be programmed into our cells, not just wear and tear. Free radical theory, 1956. Denham Harmon proposes that damage from reactive oxygen species, free radicals, produced as byproducts of metabolism, is a primary cause of aging. This focused attention on oxidative stress and mitochondria. The paradigm shift, 1990s, 2000s. This is when the optimism really started brewing. Work in simple organisms like tiny worms, C. elegans, fruit flies, and yeast yielded bombshell discoveries. Single genes rule, lifespan. Researchers like Cynthia Kenyon found that mutating just one gene, like DAF2 involved in insulin IGF-1 signaling, could double the lifespan of worms. Similar findings followed in flies and mice. This was seismic. It proved aging wasn't just passive decay, it was actively regulated by specific genetic pathways. If genes control it, maybe we can control the genes. Boom, the modern anti-aging field ignited. Elizabeth Blackburn, Carol Greider, and Jack Szostak discover telomerase, the enzyme that rebuilds telomeres. They show that cancer cells often hijack telomerase to become immortal. This highlighted telomere maintenance as a crucial aging pathway and won them a Nobel Prize. The isolation of human embryonic stem cells in 1998 and the invention of iPSCs in 2006 opened the floodgates for regenerative approaches, both for replacement and rejuvenation. So why the surge in optimism now? Because the whole ancient, burning desire for longer, healthier life has finally met a perfect storm of capability. We have the Almaster blueprint in genomics and the tools to read and edit it, CRISPR. We command the Almaster builders, stem cells, and know how to train them. We possess super senses and super tools, biotech, to see and manipulate the nano world. We wield the Y computational brain power, AI supercomputing, to make sense of the staggering complexity and design solutions at lightning speed. We have massive real-world data, biobanks, and CHAS molecular measuring sticks, biomarkers, to identify targets and track progress. For the first time in human history, we aren't just wishing on a star. We're actively reverse engineering the machinery of aging with tools of unimaginable power. We're not just hoping for a longer life. We're building the scientific foundation to make it happen. The rivers have converged. The torrent is flowing. The journey to extend our health span profoundly and significantly is well and truly underway. And that, my friends, is why scientists aren't just dreaming anymore. They're planning. But how exactly do we do it? How do we extend healthy life? It boils down to targeting the fundamental mechanisms of aging, often called the hallmarks of aging. Think of these as the key weak points where the machine breaks down. Life extension strategies aim to shore up these weaknesses. Genomic instability. Our DNA takes constant hits, radiation, chemicals, even byproducts of our own metabolism, like free radicals. Over time, the repair crews can't keep up, leading to mutations and malfunction. The fix: boost DNA repair. This is what worked in the mice. Scientists are developing drugs or gene therapies to enhance the activity of natural repair enzymes, like PARP1 or DNA-PK. Another angle is using gene editing, like CRISPR, to correct harmful mutations before they cause problems. Think of it as giving the cellular mechanics better tools and more overtime pay. Telomere attrition. Telomeres are like the protective plastic caps on the ends of your shoelaces, but for your chromosomes. Every time a cell divides, these telomeres get a tiny bit shorter. Eventually, they get too short, and the cell stops dividing or dies. This is the Hafee limit. Short telomeres are linked to aging and disease. The fix: activate telomerase. Telomerase is an enzyme that can rebuild telomeres. Most adult cells have it turned off. Strategies involve safely turning telomerase back on in specific cells using drugs or gene therapy. It's like replenishing those protective caps. Caution: uncontrolled telomerase activation is risky and linked to cancer, so precision is key. Epigenetic alterations. Your DNA sequence is fixed, but how it's read is controlled by chemical tags attached to it. The epigenome. Think of it like highlighters and sticky notes in that instruction manual, telling the cell which chapters to read loudly and which to ignore. With age, this epigenetic landscape gets messed up. Tags are added or removed in the wrong places, leading cells to behave strangely or lose their identity. The fix: reset the epigenome. This is a huge frontier. Scientists are developing epigenetic reprogramming techniques, often inspired by how we turn adult cells back into stem cells. The goal is to gently reset the epigenetic tags to a younger, healthier pattern without erasing the cell's identity. It's like carefully removing the incorrect sticky notes and re-highlighting the important parts of the manual. The mouse rejuvenation experiments often involve partial epigenetic reprogramming. Loss of proteostasis. This is just a fancy word for the cell's quality control system for proteins. Proteins are the workhorses of the cell. They need to be folded into precise shapes to work. With age, the machinery that folds them correctly, chaperones, and the systems that clean up misfolded or damaged proteins, the ubiquitin proteasome system and autophagy, get overwhelmed. Junk proteins pile up, forming harmful clumps, like in Alzheimer's and Parkinson's. The fix: boost protein quality control. Drugs that enhance autophagy, the cell's self-cleaning process, are a major focus, like rapamycin and its derivatives, rapalogs. Exercise and calorie restriction also boost autophagy naturally. Scientists are also looking for drugs to help chaperones work better or break down specific harmful protein aggregates. It's about supercharging the cell's janitorial and quality assurance teams. Deregulated nutrient sensing. Our cells have sophisticated systems to sense nutrients, like sugars and amino acids, and adjust growth and repair accordingly. Key pathways are insulin, IGF-1, mTOR, AMPK, and sirtuins. When these get out of whack with age, cells can get stuck in growth mode when they shouldn't, or fail to repair damage. The fix: modulate nutrient sensing pathways. This is where drugs like metformin, already used for diabetes, targets AMPK, and rapamycin, targets mTOR, show promise for life extension. Calorie restriction without malnutrition powerfully regulates these pathways, slowing aging in many species. The goal is to mimic the beneficial effects of calorie restriction with drugs, CR mimetics, tricking the body into thinking it's getting less food, activating repair and maintenance modes. Mitochondrial dysfunction. Mitochondria are the power plants of the cell, generating energy, ATP. With age, they become less efficient, leakier, producing more damaging free radicals, and their quality control falters. This energy crisis affects everything. The fix: rejuvenate mitochondria. Strategies include drugs to improve mitochondrial efficiency, like NAD+ boosters, nicotinamide riboside or NMN, antioxidants targeted specifically to mitochondria, like MitoQ, and exercise, which stimulates the creation of new, healthy mitochondria. It's about tuning up the power plants and reducing toxic exhaust. Cellular senescence. This is a double-edged sword. Senescent cells are old, damaged cells that have stopped dividing but haven't died. They're supposed to be cleared away by the immune system. But with age, they accumulate and become toxic neighbors. They spew out a harmful cocktail of inflammatory chemicals. The senescence-associated secretory phenotype, SASP, that damage surrounding healthy cells and drive chronic inflammation. Inflammaging, a major factor in age-related diseases. The fix: clear out senescent cells. These drugs are called senolytics. They selectively kill senescent cells while leaving healthy cells alone. Early senolytics, like dasatinib plus quercetin or fisetin, show promise in mice and early human trials, improving various age-related conditions. Removing these zombie cells is like taking out the rotten apples before they spoil the whole barrel. Stem cell exhaustion. Our tissues rely on stem cells to repair and replenish themselves. With age, these stem cell pools dwindle in number and become less active and less able to differentiate into the needed cell types. This is why healing slows down. The fix: rejuvenate or replace stem cells. This is where regenerative medicine shines. Strategies include using drugs or factors to wake up our own dormant stem cells, transplanting lab-grown stem cells derived from the patient to avoid rejection, or using partial reprogramming to restore stem cell function in aged tissues. This is crucial for organ repair and replacement. It's about restocking the body's reservoir of repair workers. Altered intercellular communication. Cells constantly talk to each other via hormones, signaling molecules, and direct contact. With age, this communication gets noisy and distorted. Inflammation increases, hormone levels decline, like growth hormone, sex hormones, and signals that promote repair become weaker. The fix: restore healthy communication. This involves dampening harmful chronic inflammation, using anti-inflammatory drugs or lifestyle changes, carefully replacing key hormones where beneficial and safe. Hormone replacement therapy is complex and requires careful medical supervision, and potentially introducing youthful signaling factors like those found in young blood plasma, though this is highly experimental and controversial. It's about clearing the static on the cellular phone lines and restoring clear signals. So, it's not one single magic bullet. It's a multi-pronged attack. Think of it like maintaining a complex ancient city. You need engineers constantly repairing the foundations, DNA repair, road crews resurfacing streets, telomere maintenance, librarians keeping the archives organized, epigenetic resetting, sanitation workers clearing debris, proteostasis, autophagy, power plant technicians optimizing energy, mitochondrial boosters, police removing troublemakers, senolytics, construction crews rebuilding districts, stem cell therapies, and city managers ensuring clear communication between departments, intercellular signaling. All these systems work together. The most promising life extension approaches will likely combine several of these strategies, a cocktail tailored to an individual's biological age and specific weaknesses. This brings us to personalized medicine. Aging isn't identical in everyone. Your genes, lifestyle, environment, they all shape how you age. The future of life extension isn't one pill for everyone. It will involve comprehensive biological age testing, using epigenetic clocks, blood biomarkers, imaging to diagnose your specific aging trajectory. Then doctors could prescribe a personalized regimen, perhaps a senolytic, to clear your specific burden of zombie cells, combined with an NAD+ booster to energize your mitochondria, plus targeted exercise and nutrition plans, and maybe an epigenetic resetting therapy every decade or so. And if diagnostics show an organ failing, you schedule a replacement grown from your own cells. It becomes proactive maintenance, not just reactive disease treatment. The developments are coming thick and fast. Beyond the mouse rejuvenation and lab-grown organs, here's a glimpse: senolytics in humans. Early clinical trials are testing senolytics for specific age-related conditions like idiopathic pulmonary fibrosis, diabetic kidney disease, and even Alzheimer's, with encouraging initial results on frailty and physical function. NAD+ boosters. Human trials with NR and NMN show they effectively raise NAD+ levels and improve markers of cardiovascular health, metabolism, and even muscle function in older adults. Rapamycin derivatives. Drugs targeting mTOR, like everolimus, a rapalog, are being tested for delaying age-related decline. Low-dose rapamycin shows significant lifespan extension in mice and dogs. Metformin for longevity, TAME trial. The large-scale targeting aging with metformin, TAME trial, aims to prove that metformin can delay multiple age-related diseases collectively, potentially paving the way for FDA approval of aging itself as a treatable condition. Epigenetic reprogramming advances. Labs are refining techniques to achieve partial reprogramming in vivo, inside living animals, safely, reversing age-related changes in eyes, muscles, brains, and more, without causing cancer or loss of identity. Human trials are likely within the next decade. Organoids. Bioprinting. Beyond flat sheets of cells, scientists are growing intricate 3D organoids, mini-organs that mimic the structure and function of livers, brains, kidneys, etc. Bioprinting is advancing rapidly, using bioinks containing cells to print complex tissue structures layer by layer. While full, transplantable complex organs are still challenging, especially vascularization, getting blood vessels throughout. Progress is accelerating. AI-driven drug discovery. Artificial intelligence is screening millions of molecules to find potential new senolytics, autophagy enhancers, and other geroprotectors far faster than traditional methods. But let's address the elephant in the room. How long do scientists have to go? When will we truly overcome the natural limits? It's the million-dollar question, and honestly, nobody has a precise answer. The 2050 prediction for some widely available life extension often refers to the first generation of these therapies, perhaps effective senolytics combined with NAD+ boosters and advanced diagnostics, potentially adding 5 to 10 healthy years initially. This isn't immortality. It's about compressing the period of frailty and disease at the end of life, pushing out health spans significantly. Overcoming the absolute limits is a different ballgame. The current maximum verified human lifespan is 122 years, Jean Calment. Could we push that to 150? 200? More? The challenges are immense. Complexity. Humans are orders of magnitude more complex than mice. Interventions that work perfectly in the lab may have unforeseen consequences in our intricate biology. Long-term safety. Proving the safety of taking rejuvenating therapies for decades is incredibly difficult. Could boosting DNA repair inadvertently help a precancerous cell? Could epigenetic reprogramming go awry? The brain. Rejuvenating or replacing the brain, the seat of our consciousness and identity, is the ultimate frontier. While neurogenesis, birth of new neurons, occurs. Replacing an entire aged brain with a lab-grown one raises profound philosophical and technical questions. Protecting and rejuvenating the existing brain is a major focus. Cumulative damage. Even with perfect repair, some types of molecular damage might be incredibly hard to fix perfectly over centuries. Could there be fundamental wear and tear that's unavoidable? Clinical translation. Moving from stunning mouse results to safe, effective, and affordable human therapies takes time, rigorous testing, and massive investment. Regulatory hurdles, like the FDA approving treatments for aging itself, are significant. Most experts in the field believe the first significant wave of therapies will arrive within the next 20-30 years, focusing on health span extension and treating specific age-related diseases by targeting aging mechanisms. Incremental gains will likely follow. Reaching 150 plus consistently might take most of this century, requiring multiple generations of increasingly sophisticated therapies. True escape velocity, where life expectancy increases faster than you age, is a theoretical possibility, but likely much further off. So, where does this leave us? Standing at the precipice of a biological revolution. The quest for longer life is ancient, woven into the fabric of our species. But for the first time, it's not magic or myth guiding us. It's the meticulous, awe-inspiring work of science. We are learning the language of our own cells, deciphering the intricate dance of DNA repair, protein folding, energy production, and cellular communication that dictates our lifespan. We are becoming the engineers of our own biology. It starts with understanding that aging isn't a single thing, but a cascade of failures. Failures we are learning to predict, prevent, and even reverse. From the tiny triumphs in mouse cages to the intricate dance of stem cells forming a new kidney in a lab dish, the evidence is mounting. Significant life extension is not just possible, it's probable. It will be a journey of incremental steps, better diagnostics, targeted drugs clearing zombie cells, therapies boosting cellular repair, lab-grown organs for failing parts. It won't be about chasing an impossible immortality, but about adding decades, perhaps centuries, of vibrant, healthy life, compressing the frailty and disease that currently mars our later years into a much smaller window, or perhaps eliminating it altogether. The natural limits of the human lifespan are no longer seen as an unbreakable barrier, but as a complex biological puzzle we are rapidly learning to solve. How long until we overcome them completely? Perhaps within the lifetime of some listening to this very recording. The science is racing forward, fueled by that most human of desires, the yearning for more sunrises, more laughter, more time. The long tomorrow isn't just a dream anymore. It's a destination on the horizon, being mapped by the brilliant minds in labs around the world, one repaired strand of DNA, one rejuvenated cell, one bioprinted organ at a time. The era of treating aging itself is dawning, and that, perhaps, is the most extraordinary chapter in the human story yet to be written. So, what do you think? Will you see the century where aging becomes optional, where swapping organs is like updating software? It's a future brighter than we ever dreamed. And it's being built right now in labs just like the ones we explored tonight. If this blew your mind like it did mine, do us a huge favor. Subscribe to Hafee Sleep for more journeys to the edge of science. Like this video if you believe in longer, healthier tomorrows. Comment below with, "Your city, Challard Country. Did you watch this with morning coffee or midnight wonder?" Every like, sub, and comment tells YouTube more videos like this. So you're literally helping us unlock the next frontier. thank you. Wherever you are, whatever the hour, thank you for dreaming with us. The future belongs to the curious. Stay hopeful, stay hungry, and as always, sleep well, dream deeper. I'll see you in the next one.